Hello everyone, and welcome to our January TNT speaker series. We're very excited to have Professor Krishna Shenoy here today to give us a talk entitled Intra-cortical Brain-Computer Interfaces from Basic Science and Engineering to Clinical Trials. So as we mentioned in the email, our goal with the TNT speaker series, especially with invited guests, is to highlight the different perspectives and ideas shaping neuroengineering today. So in that spirit, we'll spend the first 10 minutes of the hour talking about the context of Professor Shenoy's current work, a little bit about his own career and the history of the ideas that he's been working on. And then around maybe 3:20, we'll switch gears and Professor Shenoy will talk about some of the most recent work in his lab, and we'll have a second time to ask questions about that. So as a brief background, Professor Shenoy is the Hong Se and Vivian W.M. Lim Professor of Engineering at Stanford University. He received a BS in Electrical and Computer Engineering from UC Irvine and a PhD in Electrical Engineering and Computer Science from MIT. He was then a postdoc in neurobiology at Caltech before becoming a professor at Stanford. At Stanford, Professor Shenoy directs the Stanford Neural Prosthetic Systems Lab and co-directs the Stanford Neural Prosthetics Translational Laboratory, which aim to help restore lost motor function to people with paralysis through basic neuroscience and engineering and clinical trials. In addition to receiving numerous awards for his contributions to science and engineering, he's also a consultant for a number of neuroengineering companies, including Neuralink. So with all that being said, I'm pleased to introduce Professor Shenoy and let him tell you more about himself.
Great. Well, thank you so much for the very kind invitation. And it's so wonderful to see many friends. I see Nitish there and Nathan I see, and many people I look forward to getting to know more. And one day, hopefully soon, we'll physically be together again. So I'm most happy to answer any questions that you might have. I'm not sure I have anything that's that particularly interesting to share, but I do understand that developing one's career and taking into account one's interest is a very important thing. And I think that many of us have our classroom teaching in addition to our research where we try to express that. So maybe I'll just say 1 minute's worth, Cynthia, and then we'll go from there because I don't want to try to barrage you with anything. I think my particular story, just to cut to the chase, is a very familiar one to everyone on this call, probably, where I was interested somewhere between engineering and medicine all along. My dad's an electrical engineer. Medicine is what so many of us want to do because we get to help people. But how to balance between those things is always an interesting question. So I went to UCSD as a biomedical engineer, actually, and loved it. But I felt a little bit more drawn towards the electrical side. So my official degree was electrical engineering. And then I got bit by the neuroscience bug during a summer NSF sponsored research experience for undergraduates, the things that probably we all have in our backgrounds. And that caused me to really wonder, oh, maybe there's some opportunity to think in terms of engineering, but also the brain and medicine. So I went off to graduate school, wanted to really continue to immerse myself in pure electrical engineering to sort of see that through. But along the way, they had a wonderful, innocuous sounding requirement, which was to go take 3 graduate level classes in another field. And innocuous sounding because, well, how do I take a graduate class in another field? Sometimes you have to take undergraduate classes to be able to do that. That's where I met Mriganka Sir, who to this day is a friend and advisor. I went to him and he said, yes, I'm the course instructor for the first year graduate neuroscience class. Go take that class and come back and see me. And we did that for like 3 years. And every time I'd come back and my interest kept growing. And then eventually he said, you know, what you're saying is you're so interested in this, you should think of doing a postdoc. And I thought he was crazy because I don't have a PhD in neuroscience. And he said, no, neuroscience is different. It really is so interdisciplinary. So I was very fortunate, became a postdoc with Richard Anderson at Caltech, where I completely left engineering behind, well, in some proper sense, for 3 years. That was the deal, because I wanted to really jump. I wanted to really immerse myself in pure neurobiology, monkey, electrophysiology, and sort of try to become fluent, like doing years abroad to really learn the language. And I don't know. That's what you do when you're in your 20s and you don't have a mortgage or kids, or and I'm so glad I did it, because I could never do that again. And just by chance, not by design, just by chance, brain machine interfaces were just starting to be talked about. Nitish and I used to talk about these things 20 plus years ago when we first met. And, it almost sounded too good to be true, because it was sort of biomedical engineering and electrical engineering and neuroscience and medicine. But, I felt as though it was real, pursued that for a couple of years and came up to Stanford. And that's what we've pursued ever since. The science behind it, the engineering behind it, clinical trials that Professor Jamie Henderson, head of functional neurosurgery here, he and I are partners in crime, or married at the hip in that regard. And that's my story. So no grand design, simply following my interests and heart. Tried to work hard, but honestly just got profoundly lucky.
Thank you. Thank you for sharing that. So I'll take the liberty of starting off with the first question, and people can write in the chat or feel free to just interrupt after. So I guess one thing I'm curious about is your work is kind of intertwined with this whole neural dynamics conversation that's become a really hot topic. So one thing I'm curious in the history about is what was going on kind of at the beginning of the idea that neural dynamics might be a good way of thinking about the brain.
Yeah. Uh, so I do have an answer to that. And we're currently, Mark Churchland and I are currently working on a Nature Review neuroscience piece. So we can sort of write that out in addition to a couple other review articles. But basically, the motivation was that so many cell responses, or PSTHs, were being viewed at the individual neuron level. By that I mean looking at one neuron today, another neuron the next day, and so forth. And that's fine to do if the behavior is the same day over day. So no problem with that. But it was a very single neuron perspective that did not sort of take the correlation structure and the ability to look at very fine timescales of how neural population state would evolve. And the second critical thing is that the way papers were written up till around 2000 very much were to tell a story about what this neuron is doing from a representational standpoint, which makes eminent sense coming from the visual world. Hubel and Wiesel and my academic grandfather, Vernon Mountcastle, your illustrious, true, spectacular luminary there, who is Richard's advisor and therefore academic grandfather. But basically, the more you looked, the more complex these responses were, the more heterogeneous they were. You'd say, oh, a neuron is tuned, it responds more to right than left, and another neuron may respond more for up than down. Absolutely true. Apostolus Georgopoulos, again, very famous work, started at Hopkins before Apostolus moved to Minnesota. Absolutely true. Absolutely true. But the challenge was that, if you move your arm to the right faster, that may no longer be the preferred direction. So you got these non-stationarity. So every time you scratched at these motor responses and preparatory responses, the more complicated it became. And so this is really Mark Churchland and I 20 years ago. We were like, well, we don't know what to do. Let's on the one hand, be unafraid to throw up our hands and say, we don't know, we give up. We're we're young, we've got nothing to lose. And on the other hand, say, let's let the data do the talking. And that happened to be at the time where we could start recording from hundreds of neurons, where we could look at dimensionality reduction, and then machine learning techniques were starting to come along. And all of a sudden, you could start looking not at individual PSTHs as were the coveted figure ones in science papers, but instead looking at taking all the neural activity in some high dimensional space, lowering the dimensionality because the data say to, you can capture the variance, and then studying that. And that is what we would scribble on the boards, and that would lead to the next question and so forth. So it was out of necessity for just not feeling that we had enough bedrock to build on that caused us to go back to neural dynamics. And then, yeah, much to, sort of my own chagrin, many in the field are sort of pursuing this now, which I'm super happy about.
Thank you. Does anyone else have a follow-up question?
If I could, yeah.
Yeah, go.
Yeah.
Hi, Dr. Shenoy.
Krishna.
So actually, I kind of wanted to jump off this notion of recording from massive numbers of neurons at once, because of course that's, Elon Musk has been pushing this huge amount of funding into Neuralink, and the explicit goal there is sort of single neuron level control of both recording and stimulating. And I was curious to get your perspective on, so of course, Elon is not a neuroscientist. And so he's, I think, done a smart thing in that he's given lots of money to smart people to go do it for him. I'm curious what your perspective on the progress that they've made so far is, particularly as it relates to this issue of having to deal with absolute floods of data in these incredibly high dimensional spaces all at once.
Yeah, that's a great question. So yeah, I can say a lot. At some point, I hit a NDA and can't say too much more because I'm technically a co-founder and so forth and consultant. So first, I am truly agnostic to the recording modality. Something, again, Professor Thakor, Nitish, and I have talked about numerous times. I think we should explore all of them, EEG, ECOG, Nathan's work, implantable electrodes. They all have important roles. And I like to clarify that because I never want to come across as a single neuron chauvinist or something, not at all. I mean, some people will never want a neurosurgery, nor should they. Some people never want a DBS surgery or even a cardiac surgery. So, we have to have a lot of options for patients. Second, though, if we do zoom in on trying to record from large numbers of neurons and stimulate large numbers of neurons, I think we're still early enough in the game, even though it's been going on for decades, that we need a range of different technologies. Okay, so we use the electrode arrays from BlackRock, Utah arrays, as many there do, and so forth. And I'm sort of in a love-hate relationship with this, right? I mean, I know Florian Sulzbacher and Marcus well, and this is great. On the one hand, they're 510K approved. We can put them in. We know that we'll get, not single neuron sortable, but definitely great threshold crossings that are not noise for 5 years. They're safe, this type of stuff. But on the other hand, it's like, good grief, it's literally the technology I was using 25 years ago. I mean, like, what other branch of technology has stayed this static? So what Elon and the Neuralink team have done is to say, listen, let's come in with flexible electrodes. That seems to be pretty universal. Let's put mini contacts up the side of an electrode, or so-called threads, 16 of them. And then let's bring them all to an aggregator in an on-chip implantable A to D and radio system. So it's fully implantable. So that, presumably that's good. Let's put that in there. And the final thing is better have a surgical robotic, because what you want to do is do this at an optimal speed, meticulously, computer vision to avoid vasculature, all this type of stuff. So that is coming along. Pigs and monkeys, this has all been publicly disclosed. It's a long, arduous road. What is needed for FDA approval is a necessarily high bar that I defend, and but not defend against anybody, but I it has to be that way. I respect that. Now your question, Oliver, was data volumes. And, to be honest, it's not like that's been a major thrust yet. It's on the roadmap, but that's just because, until you have order 10,000 or above, what we're doing currently just scales. I mean, I don't know, just get another rack and, okay. But I think that the data processing, data storage, dimensionality reduction, model fitting in such high dimensionality, all of the wonderful data science, machine learning problems, I think it'll become much more interesting and not just, a linear build. Hopefully we can do something more than that. I also think it'll be very interesting to see how higher channel counts are justified. I'm very much against, just so you hear me say it, the arms race that often is easy to take place, because anytime you have a metric, right? Be it bit rate and some of the things we do, or neuron count or electrode count, it's easy to just say, oh, I want another order of magnitude. Well, why? How are we going to demonstrate that 1,000 really is better than 100? Because 1,000 is not better in terms of surgical risk. So is that going to be because of robustness? You could have 90% of the electrodes die and still work? Or is that going to be that you can perform much more complex multidimensional reaches, including sensory feedback, as you guys are working on, along with APL and stuff? I think it'll be interesting to see what those domains of crisp advantage really are.
That was a really great question. And maybe if we have time, we'll come back to all of these things. But I think because it's 3:20, we should let you transition over to talking to what you're working on right now.
Okay. Thanks. This is wonderful. This is a great format, by the way. Okay, so here's what I've tried to do. So I've taken a little bit of liberty and I'm deviating from the title slightly. It's still the same spirit. And the reason is that I think on Zoom calls like this that we're all getting better at, I suppose. But it's better just to sort of like get down to the point as quickly as possible, because it can take a little longer to have questions come up and so forth, and make sure that something of meaning is said, as opposed to trying to review too much. So I stripped it all down to our two most recent human projects. So I won't be talking about the basic science and the neural dynamics. It's under the hood and used, but I won't give that talk. And I'm not going to do any really, sort of what have we done in the last 5 years. This is all like last couple of years. It's just come out in print or about to come out in print. Okay. So, with the goal of hopefully that's the most interesting. Okay, so can everybody see the title slide here? Okay. Hang on. Yeah, okay. So I will very briefly, because I think you're all quite familiar with it, describe a brain-computer interface where we often append intracortical, just to make it clear that we're talking about, sticking electrodes in the brain, right? Then I'll describe a handwriting BCI and tell you what that's about. But basically, we have a person imagine moving their hand to print out letters on a notepad with their hand. Now they're paralyzed, they can't move it, but they can think it, they can attempt it. And we can decode that and then type out letters on a screen, something we didn't really think would work or didn't know if it would work because it's decoding very fine, dexterous, high-speed movements. But I'll show that. And the second area is speech. Mark Slutsky, Eddie Chang have done beautiful work in ECOG, and others, of course, have done beautiful work in ECOG. This is the first that we're aware of single electrode and populations thereof in the same hand knob area where actually speech by sort of a strict homunculus definition shouldn't exist there, but we find it, and then that can help us guide where to put our electrodes next. And we have not demonstrated this all in closed loop real time, but we've laid the foundation for it. Okay, so handwriting and speech, and we're done. So we are all on the same page, I think, that what we want to do is help somebody that can no longer move due to a spinal cord injury or neurodegenerative disease. We like to help them by moving a prosthetic arm to pick up that cup, stimulating the paralyzed musculature, functional electrical stimulation, or go ahead and move computer cursors on a screen or interface with computers, which, you know, today versus 10 years ago is an awful lot of our days, right? If you actually sit down and think about how much time we actually just spend in front of a computer, it's, well, it's a little scary, but okay. And the way we can do this is we can rely on people still being able to see where they want to move and process that visually, formulate plans and elaborate those plans in the premotor and motor cortex, okay, and send those signals down. But instead we can put our electrode arrays and intercept these signals and do these different functions, okay? So again, there's huge networks here. Subcortical structures are fascinating. It's not all about the cortex, but that's where we focus. Now, the new directions that I'll be describing, okay, are to investigate what I call human-only behaviors, okay? And this is sort of one step beyond what we've spent, you know, previous years doing, which was, hey, we learned this in the monkey about motor movements. We therefore have a pretty good idea of how it should work in humans. Let's go do that. And we've done that. And that, I think, was very exciting to all of us because it established the intact monkey model as a good model for people with paralysis. Okay, I mean, nothing is perfect, but it translated. It's not like sometimes you run into, you know, curing lots of mice of cancer, but it doesn't work in humans right away. And we're going to be using high bandwidth access, meaning action potential access to large populations of single neurons, okay, as we've already talked. And we want to increase the performance and the capabilities of these systems. And we're investigating really rapid and complex motor sequence. That's what handwriting and the motor act of controlling the vocal cords and controlling the articulatory pathway have in common, okay? And this beautiful graph by Eddie Chang and Gopala really show us where we're at, where, you know, we're way down here in BCI-driven cursor control. Maybe handwriting can come up. And then, you know, all the way out here is conversational speech and so forth. So, you know, we're in no danger of going, saturating what is needed here. Okay, so what about handwriting? So this is the work of Frank Willett, shown down here, brilliant postdoc. And what he did was the following. Have our participant imagine a legal notepad just to set a size scale, and imagine holding a pencil, and imagine writing out in not cursive, but close to cursive, you know, just sort of print and just the natural writing that he would do. No special training, okay? And after, a couple sessions, just to make sure he could do that consistently, which is not easy because you don't see your hands move. He is paralyzed. He can't move. Okay. And we record from, in this case, the arm and the finger areas of the brain. So here we ought to have pretty good information about these, except that these are really fine movements, right? This isn't a gross arm movement. This is, tiny coordinated movements of the fingertip that are amplified over the length of the pencil to get that, okay? And then the goal, very importantly, I want to clarify, the goal is not to recreate this pen stroke, okay? That the H looks like this and the E looks like this. One could do that. That would be like pure motor decoding. Instead, we're interested in classification. We want to see if he can write this hello, because that's fast for him to think of doing, but classify as an H and then type just a standard H up on the screen, okay? And what we are going to do is wait for a letter to complete before we look backwards and classify what that letter is. Was it a space? Was it a letter? And that's why you see hello is completed here, because the O is complete, but the W isn't quite complete, so we're waiting. Okay, so we'll have a 1-character latency. All right, this just shows on MR image of our participant T5, who has an upper spinal cord injury incurred about 9 years ago. And the electrode arrays are in the hand, finger, so-called hand knob area. The sort of the place that we, as part of BrainGate with Lee Hochberg and John Donahue, always go. Okay. And we got started with this task shown at the bottom. We'd say, please prepare to write an A. Okay, we'd wait, and then we'd say, go. Okay, now imagine writing that A. And then there'd be stop. And okay, now start preparing the next letter. And we go letter by letter. Okay. And we do this for, the first day was like a half hour. Okay. Data sufficiency, because this is all powered by machine learning, is a thing. And I'll tell you the tricks to this. Okay. So we do this character by character. Okay. And then again, we're not going to be doing this for the real system, but we wanted to ask, can we actually reconstruct the imagined pen tip trajectory from this neural activity, just as an existence proof that we're getting well-modulated signals? So we just build in this case, just a simple, Kalman filter where we get the tip velocity as some linear function of the firing rate vector, okay? And here's what we get on average, okay? For an A, you can see something that looks like a B, B here, C here, D here. Other letters can get a little bit more confused, but if we take what I just showed you there on single trials and then cluster it in a 2D space, t-SNE in this particular case, but it's about the same MPC, we can see that we have good cluster separability for the 26 letters and the 6 special characters, okay? And we can classify with 94% accuracy. So this is sort of like, okay, the game is afoot. We've got modulated signals. We seem to be able to classify them on a single trial. Now let's go build the system. So what we did is say off these 2 arrays, we'll have about 192 electrodes, exactly 192 electrodes. We are going to set a threshold crossing at minus 4.5 times the root mean square of the voltage. And anything that crosses it we'll call a threshold crossing, which is largely 1 neuron or potentially 2 neurons. So there's no claim that we are spike sorting, but absolutely we feel as though we're getting spikes as opposed to noise, okay? And not low frequency local field potential. All that may be even more useful. I'm just describing what we did here as a starting point. Okay, now in a narrow time band as we progress along, we go through a recurrent neural network, okay, that we train. How do we train it? Well, remember we had all that training data from single letters. Okay, We also had training data from when we put up sentences and he wrote out those sentences, okay? And third, and really critically, what we did was we then took all those individual letters and the letters as embedded as part of sentences and cut them out and then reassembled them synthetically, putting them in all these different orders to generate even more training data. That's the key. That way you are not data limited. You don't overfit or underfit your models and you get good generalization, okay? Because this is always a problem with ML hits neuroscience, right? Neuroscience is not as big as all things you can search on Google, and therefore you can't do natural language processing as, swiftly. So the probabilities for each character you can see in real time here come up and down, okay? And then you also have a new character signal. So it rises high when it feels though it has a new character, okay? And goes back down, new character. So this is like a clock signal that tells you when you want to read out the different letters. And instead of a space, we put in a greater than sign because a space is very hard to differentiate from a pause. So if you want to separate your words, you write a character. So that's, that's not totally normal, but it's like not hard either. Okay, we did online thresholding. Just imagine a threshold along here, and we get the raw output. And in this example, you got TNE paper instead of the paper. That's a mistake. Well, the beautiful thing with English language is it's a mistake, but you still know what it means. Okay, so we could be satisfied with that. We're not. Okay, we want to keep getting that error down. Okay, that single character error. And mercifully, as I'll show you in the next slides, that error rate, which I'm showing you to be clear that there are errors, it turns out it's actually really low, even on its own. But if you want to clean it up even more than what we did offline, we did not implement this yet online, we're in the process of that, is you can also run through a language model. Okay, this is just natural language processing on a dictionary. You know that if you have a T, you have certain transition probabilities through the dissension tree. You can then optimize with Viterbi search, and then you can have a retrospective output language model where you could have the paper. Now, how would this work? You go through TNE space, then this model would be assessing and going, that's not right. TNE is possible, but weird. It's much more likely it's THE, so you could just have it autocorrect that whole TNE to the, okay? So there's a whole, there's a huge number of ways you could go with this. This is an engineering design space now. Okay, so what I'm gonna show you now is the raw data though. Okay, the raw signal, because we can always clean up from there. So what we did was we ran this for 5 days in a row, okay, in this data shown here. And the characters per minute, we were able to get up to about 90 characters per minute. Okay, now what's the error rate? So for the same data across the same 5 days, in the circle is the raw error rate. So here it's about 5. Here, this is bad. This came up to like 12%, but this is like 7%. But the point is that it's pretty much in the, I don't know, 7 or 8% or less. If you add that language model, which you can totally fully run in real time, it really crushes them down to about, on average, 0.5%. Okay, so there will be errors, but it's so low that it is, we think, a viable communication prosthesis. Okay, and the character error rate I just showed you here, this is with language model, and word error rate is over here. So how does this look? So I'll play this movie. We'll see if I get lucky and it plays. Okay, so he is asked to copy the sentence up here. And all that's happening is that letters are appearing somehow magically out of the ether. But that's because his right hand that you can't see, but we've got another camera on it to document that it's not moving. He's imagining writing, and this is all real time. Sorry, the jerkiness is just because of the bandwidth. Okay, now let me show you that video one more time, so it'll be here, and you're seeing 90 characters per minute with over 95% accuracy, about 15 words per minute in real time without the language model. It can be better. And right below it, we put what we think is the previous record, which is our own, from the Pandranath and Nujukian paper, okay, where a cursor is moved out to a QWERTY keyboard and selected by imagining a click. And it just so turns out it's the same participant. This method was done 3 years earlier. The signals are quite comparable. In fact, you'll see this go way faster. And we actually use the exact same sentence just to make the point so we can do a head-to-head comparison. And the videos are very precisely aligned. So here you can see sort of the classic 2D cursor plus click method, which works just great and has big advantages. You can go into continuous draw mode. There's lots of reasons for this. You want it too. But up here, if what you're wanting to do is just communicate text, it's 2 and close to 3 times as fast. And so to our knowledge, across any modality, including SVG, it's faster. Okay, so that completes handwriting. So we want to keep building on that. And we also very much like to try to get systems to the point that they're robust. And then, then we move on, we're at university labs, so then we move on to something basic again, because otherwise, we're not as good. We're no good at making products, right? So, okay, so what about a speech BCI? So what's the setup here? Well, I don't know. First of all, speech is cool, okay? And we all know that because, I think we all probably had our brief scientific love affair with birdsong, and, it's just awesome. And human speech is just, a pinnacle of ability, of course, at some level. But it's human only. I mean, it's really hard to have an animal model that comes close to volitional speech, okay? And this real-time MRI, which is not ours, I don't know, I just think it's cool because, look at the tongue, look at the cavity, you're controlling breathing. Look, this is a really complex motor control problem. And as a motor person, that's intriguing. And we know that people can't speak. For example, towards the very tragic end of their life for Lou Gehrig's disease, ALS, amyotrophic lateral sclerosis folks. And so Sergei Stavisky and Frank, Guy, Sergei, who's wonderful, everybody's wonderful, Guy, and then Shaul Druckmann, who's a colleague in neurobiology, previously of Janelia, okay? We set to work on this, and this is all with Jamie Henderson, who I'll have a special shout out on the final slide. And so we had people, in this case, our participant could speak, okay? He had not yet lost that ability. So it was a unique opportunity to record while speaking. Now, how is this possible? Well, we did not go put electrode arrays in IFC or Broca's area, okay? We had arrays and we were opportunistic. We had them sitting right here in, this is an artist rendition, so it's not super accurate, but it was sitting in hand knob area, the exact same array locations as before. So it really should be far more lateral or ventral before you get to speech areas. But, welcome to the wild and woolly world where different functions are really where they shouldn't be and back and forth. The signals are undoubtedly weaker than if we properly targeted them. And that's what we're going to try to do with our next participant. So they would be instructed to say words, would record that auditory track, would record the neural activity, and then we'd see how far we go. And we're really inspired by the beautiful work that Eddie Chang and Mark Slutsky and many others have been doing with ECOG grids. But of course, ECOG grids are recording from many neurons at a time from the surface as opposed to single neurons. So our participant heard a couple of beeps, a phoneme or word was played, a couple of go clicks saying, go ahead and get ready. And then this is voice onset. He started saying ba, and we recorded ba. Okay, and that's our data. And then we can go look at the neural activity in relationship to this voice onset. Okay. And if we look at our 2 electrode arrays, these darkened arrays are where we found the modulation for speech. So they're really spread out quite a lot. This is all precentral gyrus, okay? This is 4 millimeters by 4 millimeters with each electrode spaced by 400 microns. And if we just take an example neuron where we actually did spike sort it to make sure that we were picking up on single neurons, okay, later we'll not spike sort, but we always like to cross-check. That neuron's raster plot of repeated trials when he was saying nothing, repeated trials when he's saying I and you and ba and ga, you can definitely see that it's modulated, but of course it's Poisson spike, so it's a little hard. So here's a PSTH. And here you can see that red and green, something in here is much higher than silent and much higher than, say, purple, shh, okay? And this modulation is happening between the time of the go and the voice onset and then during voice onset. Now another neuron over here has a different pattern, okay? And a natural question to ask then, let's take a look at I here, okay, is what electrodes are modulated when saying the phoneme I? Well, all these colored in blue. So this is a very sort of same motif we often hear, right? It's a very distributed code. Many, many neurons across many millimeters, probably, actually even across centimeters. And this is probably a centimeter dorsal or medial to where you even ought to find speech motor cortex. This is all motor, right? We're not talking about actual like conceptual language. We're talking about the production of language, right? Okay, and we can do the same for the other phonemes. And we see that there's different patterns, but in all cases, they're quite distributed. So most functioning electrodes modulate to multiple syllables. And on average, any given electrode is modulating to 4 of these 9 phonemes. Okay. That's one participant, T5. T8, his arrays are not quite as strong, but we see the same trends, the same tendencies of distributed codes. Okay, so this is 2 participants. This is possible because of the BrainGate clinical trial where we work closely with Lee Hochberg and John Donahue on the East Coast, and we run each other's sessions and talk weekly and all this. Okay, now we can also ask for an individual neuron and for the population as a whole, if modulation is stronger while listening, is this auditory or is it the actual production of speech? So we aligned to the onset of hearing the sound when we actually played spoken language to the person, but didn't ask them to try to speak, versus speaking, and it's very clearly speaking. Okay, it's not a reliable response in an auditory sense. It is in a speech production sense. Okay. Now, I've been claiming this is related to motor production. If that's true, these neurons had also better be modulating when we just have the person open and close their mouth, but not saying words, or lips, or tongue, and so forth. Now, it's harder, of course, to ask them to flex the muscles of their vocal cords. But what we find is neurons that are indeed modulated. These colors correspond to the colors of the titles up here. Okay. And what we find across both subjects is that some electrodes respond only to syllables. That's in red. And some respond only to these orofacial movements. That's in blue here. But most respond to both the syllables and the orofacial movements. So if you add purple and blue, what you're seeing here is that the vast majority of cells are motoric and specific, specific to the effectors that would generate speech. Okay, and this seems very consistent with the other things that Chang group and Slutsky group and others are saying. Okay, so final few points. Intra-cortical recordings seem to be revealing speech-related activity in this hand knob area, not more lateral. Sorry, I shouldn't say not more lateral. We did not measure more lateral. We would expect to find these types of responses and even stronger if and when we go more lateral. But we're finding them where we sort of shouldn't find them. Is this due to remapping due to tetraplegia? Well, we don't really think so, due to another study that Frank and Daryl headed that came out recently, and also a study by Richard Anderson. And that is these electrodes, while they are modulated by the facial muscles that I described, so this is 2 standard deviations of modulation, so it's well modulated, nevertheless do modulate even more for sort of what it should be modulating for. So it's consistent with Penfield and Wilder. If you stimulate on the surface, yeah, that's the arm area of cortex, but it doesn't mean that that's all it's doing or that's all the signals are doing. Okay. And it is consistent with ECOG, where you're measuring the strongest signal, but there's still lots of other signal. And this really fits into this major theme in systems neuroscience of mixed cell activity, right? Okay. So single neuron, the single neuron motor map appears to be more distributed and overlapping than seen with ECOG grids or electrical stimulation. So what am I really sort of trying to say and sort of not trying to get in trouble saying, I guess? I don't know. I mean, I'll stick to the data and I'll feel comfortable then. You know, we all get that the literal textbook, motor homunculus, which has our rays being in this area and therefore should be hand, is not literally true. Even Penfield recognized that. So I do not mean to be saying at all, oh, look, it deviates from this map a little bit. That I think that ship sailed decades ago. Okay. Instead, it's just an appreciation for how distributed this is. That we could put arrays across centimeters of cortex and pick up on various levels of speech-related activity. And this could be very important for designing and maybe minimizing risk and invasiveness, speech prostheses. Okay, last decoding. We can go in, we can look at the clustering of different phonemes. This is decoding single trial neuroactivity, right? Because I have not shown you the ability to decode single trials yet. Is the signal that I've been describing is weaker than it ought to be in another area? But is it strong enough that we could decode it on a single trial if we wanted? The answer is pretty much yes. Not great, but yes. So we had 420 different words sampling from all phonemes, okay? And we performed an experiment where we had all the spoken phonemes and all the decoded phonemes in multiple hour data sets, and then laboriously hand labeled where voice onset was and so forth, just to make sure we got it right. And overall, we could get about 29% correct accuracy across all 39 phonemes, which is, it's not 100. It'd be great if, as we go more lateral, we start pushing into the 80s or 90s or so, but it's way, way above chance. And actually, this number is already pretty similar to ECOG results from across, large swaths of the brain. Okay, the final steps where we'd like to head from here, we've not done this, we're in the process of doing it, is of course to do this in real time, to have the person say something, record the neural activity, chop it up and predict it, okay, and then play it out of voice synthesizer, not unlike what Eddie Chang's group has recently done with ECOG. And of course, the contention is, we haven't exactly, we informally we think it's true, but we haven't formally proven it. Just like a person who is paralyzed and can't move their arm, they can still think about moving their arm or attempt to move their arm, and the signals seem to be quite similar. So too, after a person loses the ability to speak, presumably they can still attempt to speak. So we would imagine that even when the actual recorded audio is a flat line here, we would still get modulation according to the attempted speaking, like the attempted handwriting. We don't know. Well, that'll be a nerve-wracking couple days when we try it. Okay. And of course, let's not just put electrodes here, or we don't have to put them there ever again, but let's go more lateral. Sort of basically by opportunistically already having electrodes here and finding something, it's gotten us more enthusiastic about trying to go more lateral, ventral premotor cortex, or ventral precentral gyrus, maybe IFG, Broca's area, that range. So in conclusion, we think that handwriting is a way to provide robust neural representation of a highly dexterous behavior after paralysis. And we demonstrated in real-time handwriting that has a pretty high characters per minute rate and accuracy. And importantly, this BCI is entirely self-paced. The person could write, take a break, write on their own. There was no metronome clock. It was not a clocked system. So it's most natural. Behaviors that are time varying may be easier to decode and lead to higher performing IBCIs. I did not have time to go into this, but in the bioRxiv paper, the question is there something about making hand movements that make them fundamentally easier to decode than straight movements? And the answer is yeah, there is. And it's the temporal complexity. And so we have some simulations and computations there. Okay, speech BCI, there's broad tuning for speech. We've observed speech for the first time in dorsal motor cortex. These neuron signals show promise, we think, for speech BCIs. And finally, the most important slide I ever show is the acknowledgements, the people that actually do the work. Guy and Daryl and Sergey and Frank and Donald, who do all of that. Lee, of course, as part of the clinical trial. This is Jamie Henderson. Special shout out to him. We've worked together super closely, sitting next to each other for the last 12 years. So he's the hands and the brains. I'm just sending emails, I think. And Shaul, who's just a brilliant computational neuroscientist and previous alumni and our funders. Thank you.
I'll clap for everybody. And we have a ton of questions, which I think I'll write down and potentially send to you because there's no way we can get through all of them. But
Sure,
one
I'm happy to try to respond. Yep.
Oh, thank you. So the one that might span both of them and a few people's question is there's obviously some history into hand motion. So the way I described it was like if you write the word previous, the R and the E change the shape of the E compared to the E being at the beginning. So there's some context to your letter. And
Yes.
similarly in speech, there's some context to your mouth movement.
Yes.
So one part of the question was when you make these decoding algorithms and you've mixed up the letters that you now took out that context of the order that they wrote, is that affecting your training? And similarly, should we care about that when we move over to speech?
Yeah, so that's a great question. So co-articulation is the I think what's being talked about for speech, that it very much matters what the preceding and even the next phoneme will be. That will be, I think, very important for speech. For handwriting, we don't know, but what the analog to that would be is if the current pin location leaving one letter it forms a different starting point for making the next letter. So that would presumably be the analog. And the way we've either cheated, but not too terribly explicitly cheated, is that by printing, not cursive, but by printing, you usually make a letter and come to a stop. And you come to a stop. So that coming to a stop, and usually down at the bottom of a line on a ruled piece of paper, if you thought of it that way, is probably what breaks that dependency, that ordering effect, that co-articulation. But to go faster, writing in cursive, I bet co-articulate, and we have discussed it. I mean, I think co-articulation probably has a handwriting analog.
Another one that's interesting, if you have time, was someone was asking about the user experience. So in terms of workload fatigue using this interface, did you observe any evidence of adaptation either at high level, maybe the user consciously adapting their writing strategy, or low level changes in the neural response as they use the interface over time?
Yeah, that's a great question. And we, currently still have very limited data to really get at that level of question. But we do have some sense of things. Okay, so first of all, I think the overall cognitive load is very low, as evidence to the fact that so our participant T5 is very hardworking. We often have to get him to stop, so it's worth saying that. Okay, and that's not always the case, as many of you know. Okay. But with that said, knowing him for a few years, he could readily do this for hours a day, let's say 3 to 4. Enjoyed it, didn't find it hard. Now, what did he try to do? What he tried to do, and this is not like we do a double-blind study or something, we're working with them. We say, listen, the deal here is the more reliably you write the letter A, the easier it is for our algorithms to pick up the letter A. The more reliably you write the letter B, same thing, and so forth. So please try to write reliably. We don't really care how big it is or what style it is. Just, choose what's comfortable and what you like, what you know, stick with it. So I do think that day over day, the fact that we saw improvement had to do with that, as opposed to true adaptation or plasticity at the neuronal level. But what would be very interesting is to sneak in a perturbative adaptation experiment. So, get him going on this, and then just have it be that we perturb the path on every letter L, right? and so he makes more mistakes on the letter L. It's confused. How does he adapt to it strategy wise? Is he conscious of it? How does the neural activity change? That's nothing insightful or unique. That's just sort of a standard toolkit thing one would do.
Do you have time for any more questions?
I do. I'm okay on time for another 5-10 minutes, but I don't mean to be disrespectful of other people's times. I apologize for running 5 minutes longer than I had intended.
No, I mean, it was great. So I guess just in case people start to disappear, thank you so much for coming and talking to us, and we really appreciate it. And everyone's
Thank you all.
free to go. So
It's, in this day and age, it's just great seeing friends' faces. Yeah, sorry, go ahead, Cynthia.
so I'll pick a few more from here that look interesting. Let's see. Let's see. So here's a recent one. Could circuits for motor cortical modulation of auditory cortical activity, circuits for cancellation of self-generated speech based on sent motor command, for incrementing the quality of separability of speech data. Wait, I have to reinterpret that. Giorgio, would you want to just say your question out loud? Sorry.
Yeah, sorry. I forgot to say be used. I meant, I know in the auditory system, there are some circuits that, based upon the incoming motor command, generate a kind of a copy of the speech that you are expected, the speech sound that you're expected to receive.
Yeah.
And so, so then our brain can cancel from all the incoming sound, like the speech generated sound. So could those circuits, like the signals coming from those circuits, which are related to the speech, by the intention of having that speech be used, to have additional data recording or
Yeah, so I think it's possible. So just to make sure I understand it, if I want to say a word, then a copy of that outgoing signal is made and run through what in the motor community is typically called a forward model.
Exactly. That's what I'm referring to.
Yeah.
Obviously, I'm not an expert on this, so it was just like,
No.
an idea.
I'm just speculating too, but hey, it's a Wednesday afternoon. Okay, so a copy is made, it's run through a forward model, and the sensory consequences of speaking are thereby estimated at very low latency, right? And that can be used to subtract out the signal that auditory cortex would otherwise respond to, because you don't want to hear yourself as much as you want to hear the other speaker. So if we knew anatomically where that signal you're talking about, the feed forward signal is, then yes, we could put, hypothetically, you put another array there that would have good information about what you want to say. That would be another source of information.
Okay, I was just curious. Thank you very much.
Yeah, I think that's I've never heard that. That's a great question. Yeah. I mean, while we're at it, I think that, the same should be true for motor control. And I don't know, I think cerebellum is such an interesting place that maybe you should rope Reza into this question, see if, something in cerebellum would be fun to work with. Yeah.
Yeah. Thank you very much.
Mm-hmm.
Another good question was there any way for patients to indicate that they'd made some type of error and delete something that was written?
Yeah. So no, there was not. So it was a closed loop handwriting experiment, right? Because he saw a letter, he brought it in, he planned what letter to make, we read that out and we put it up on the screen. But it wasn't like there was the ability to delete that or so forth. Now, I think there's two ways to expand this. One is a delete key, right? So some other syllable that says, I don't want this syllable to be typed there, but some, I don't know, some like less than sign that says delete the previous key. I mean, it literally is the delete key on your keyboard, okay? We devote a key on a keyboard to delete, okay? The other way, which is actually much more intriguing, of course, is to detect an error signal from the same electrode arrays, if possible, which we have found, or from some other part of the brain. And clearly, when we type a letter that's wrong, we know it's wrong. That's what causes us to hit the delete key. So why wouldn't you, if you had access to it, read that signal out of your brain and just rapidly delete that key and save yourself the whole time and effort? So that's that automatic error detect and correct type system could be appended to many different types of communication systems. And that's sort of, I don't know, I've always been fond of that because that's something I can't do with a regular keyboard, right? Everything else is trying to restore the ability to work with a keyboard as you used to be able to. And while I'm not at all big into augmentation, doing things that are way beyond human abilities at all, in this case, I mean, yeah, what's the harm?
If you have time for just one more, this is a little bit more out there question.
Sure.
But I noticed that for speech, decided to use a phoneme type decoder, and kind of in ML, usually people have found that if you just give natural speech, a deep learning algorithm will do well. So I assume you used a phoneme because the human brain and speech apparatus works with phonemes. I guess the question just generally going from there is, do you find that you think for brain-computer interfaces we'll have to work with the way that the brain thinks about information? Or do you think you're going to find that we have to kind of just do whatever works with these algorithms?
Yeah, I think that's a great question. It's and I call my well, maybe not quite so PC, but I call myself a happy schizophrenic. Not that I am. The reason I say that is the following. I'm very happy to work on basic science. I do want to know how the brain actually does it, phonemes and so forth. But I'm also a very happy engineer where it's not that I don't care, but I want to build the best system that works the best possible to help this person. And the reason I'm happy with that duality or those two things is that any project can be one or the other, but I don't insist on them being both. In fact, it's very, I don't know, I think maybe we've done 2 or 3 papers where I'd say, okay, actually, this is sort of 50-50. And part of that comes out of a fear of sort of splitting the difference between the two and not quite doing science and not quite doing engineering, right? I think everybody, all of us that have worked on anything interdisciplinary are familiar with this feeling and being on guard for this, right? So back to your question. I think that we'll want to do both. We'll want to see if the basic science understanding can help us do better in brain-machine interfaces, but we shouldn't not build the best brain-machine interfaces using modern machine learning, operating on words or beyond that with whatever models we want. So both is my cheat. My answer, A or B, is A and B. Yeah.
Okay, well, thank you. I think that's covered a lot of the questions and it's a pretty good place to stop.
Great. This was super fun.
Yeah, it was really great to have you. Thank you so much. I'll stop
Thank
the
you.
recording.
Well, thank you for